my name is Rachel Whittington. You guys might have seen me around at some meetings or maybe at your properties, but I'm the GreenCorps member with Riley Purgatory. I started in mid-September of last year, and I will be here as a GreenCorps member through mid-August. And today I'm going to tell you guys about Minnesota GreenCorps as a program and also some of the projects that I've been up to over the course of my term. So Minnesota GreenCorps is an AmeriCorps program, and AmeriCorps is the big nationwide network of service programs that individuals participate in every single year. And those programs can fall into categories such as public health, education, safety, and environmental stewardship. So Minnesota GreenCorps is a Minnesota-specific AmeriCorps program that is designed to help communities build climate resiliency in communities that need it. So we serve throughout all of Minnesota. We serve primarily in the metro, but also in greater Minnesota, such as Southeast and North Minnesota. And GreenCorps members are assigned to host sites, and those host sites can be any organization such as a local government unit, like a city, a council, a county, a watershed district, can be a state agency, a school district, or a nonprofit. And going forward, a lot of host sites are aiming to target environmental justice and environmental justice initiatives, and we have about a 60-40 split between sites that are in the metro area and sites that are in greater Minnesota. So along with doing projects throughout the year, Minnesota GreenCorps is really great about having program support. So we are provided a $300 budget throughout the course of our term that is required to be provided by host sites for any sort of trainings, certifications, or conferences that we choose to attend during our term. We also are provided a mentor throughout our term who we meet with monthly to discuss progress on our projects, any issues or concerns we might have about our host site, or anything else we just want to chat about, and also just have somebody to talk to who is a bit more familiar with the field than we are. And we also have an alumni association that we can engage in post-term as well. So Minnesota GreenCorps is separated into 4 different topic areas, and you can serve in topic areas such as community outreach, energy conservation and transportation or air pollution reduction, stormwater and forestry, and waste reduction and recycling. I am in the stormwater and forestry group, and yeah. The activities that members take part in, it can include anything from any of these bullet points. It can be implementing projects, engaging the communities through education, and it can be assessing any type of conditions through data collection or analysis or benchmarking. So each member is also given a work plan that helps guide what they do throughout the course of their 11 months. And I boiled down my work plan into 3 major project areas. I boiled it down into education and outreach, which is that picture on the right over there, data collection, which is the bottom left picture, and database administration. And that is a picture of the database that I have been working on revamping over the course of my term. So I'll talk about data collection first and kind of explain what my role has been in that and what exactly we have been collecting data for. So we've been collecting data for the soil health study that is being done by the district. And that aims to answer 3 main questions. We want to see what the current state of soil is in our district. We want to look at how that could inform soil health metrics in non-rural settings and how it can inform future restoration sites within the district. Data collection for the study started in around 2022 and is going to continue into the foreseeable future. We do 3 main tests for data collection. From left to right, we do soil profiling, infiltration testing, and sampling. And I'm going to talk a little bit more about each test that we do. So profiling is looking at the horizons or the layers in the soil. We typically dig about a 24-inch deep hole into the ground, and we analyze the horizons in within that 24-inch piece. And those horizons can tell us about what minerals are in the soil, texture of the soil, and how that could affect infiltration and how it holds water. And could tell us how much organic matter is in the soil as well. So that picture on the left is some of the instruments we use there. The gray tool on the left is an auger, which we use to manually dig that 24-inch hole. We also use a Munsell soil coloring guide, which is that picture right in the middle there. We will take a piece of soil from a horizon and get it a little wet, and then indirect sunlight, which this picture doesn't show, but indirect sunlight, we will look at that piece in direct sunlight and try to color that piece of soil with the color that it best matches in the book, and we will record that. And then we also do another test on site called texturing. So this is to help make sure that we know that each horizon is unique. One of the telltale signs that a soil horizon is different from one another is texture differences. So we categorize that as it being smooth, gritty, or somewhat of a mix in between. So that's what that picture is showing there on the right. That involves taking usually a little piece of soil from that horizon and getting it wet enough to where you can feel the texture. And then you'll be able to feel once all the organic matter is gone, whether it's smooth, gritty, or a combination of both. The next test that we do at most of our sampling sites is an infiltration test. And for this infiltration test, we used a modified Philip Dunn infiltrometer, which is a glass tube that has a head, and when you fill it with water, it will take data points as the water is infiltrated into the ground. And with this test, we're specifically looking for a KSAT value, which is not the infiltration rate. The infiltration rate is how fast water will infiltrate into the ground. The KSAT value is how fast water will infiltrate once the ground is completely saturated. So, and then there are some pictures on the right of our KSAT in action and the case that it comes into that we pull around to all of our sites. The final test that we do is sampling. So again, the picture on the left shows some of the equipment that we use. The shovel, we keep our samples in a cooler to prevent further decay of soil once we take that sample out of the ground. We have a bucket to help mix, and we also have Ziploc bags that we use to carry our samples. So we take pretty small portions of soil out of the ground. We only take 6-inch by 2-inch chunks for our subsamples, and we use all those subsamples at a single site to create a composite sample. Once we have that composite sample, we will send that on its merry way to the soil health lab at Cornell University, who analyzes that soil sample and will give us a lot of different metrics that we can't test for on site here. And once they do that, they will send us back a report that has all of those metrics categorized into physical, biological, and chemical changes, along with the texture class of the sample for that specific site. So for example, this is classified as a sandy loam, and it has the breakdown of all the soil particles, and then it has the values that the lab found for each of these metrics, along with their comparative ratings. And during the course of my term, we have been doing there are two main projects that I have been working on. The first one has been a residential lawn project, which has about 20 total sites and has been sampling residential lawns that primarily have turf. And the folks who have given us permission to do this project have been members of our Board of Managers and members of our Board of Managers and Citizens Advisory Committee. And these are a couple pictures I have pulled from some sites. So this picture right here is from one of our staff's folks' place. And then this place is from, I believe, from Andrew Eller's place right over here. Just to give an example of kind of what some of these sites have looked like. And then this project is going from April until late June. And then the project we'll be working on for the rest of the summer is a Minnesota Arboretum project. And this project is really interesting because we're actually going to be sampling a lot of different ecotypes other than turf. We'll be sampling ecotypes such as restored and undisturbed prairie, different types of woodlands, and old field. And we are planning on sampling at the Arboretum from June through late August. And that's about all to say for the data collection project. Next project I'm going to talk about is kind of what I like to call the database administration project. So we need a place to put all this soils data because without that, it would just kind of be a huge mess. So we have been creating a Microsoft Access database to house and generate site-specific reports for all of the soils data that we sample within the district and the adjacent areas. As of right now, we have about 30 sites, and the database measures for about 67 metrics for soil data. And we use Microsoft Access and then Visual Basics for application to run the database. And then to give a little bit of a timeline as to how this database was created, from September to November, I used Elena. Last year's Elena was the previous GreenCorps member before me, and she made a database in Microsoft Access that wasn't completely finished, but she sent over to me when I first started to get an idea of what this database would look like. So I spent a lot of time working with that database and messing around with the code and design to figure out how I could get it to work. And then I also researched and learned a lot about basic coding and design techniques in Microsoft Access, since I didn't have much previous experience with Microsoft Access. And then I also was able to take some classes through LinkedIn Learning to further my knowledge about that. And then in November, I went around asking people at the MPCA, or specifically people in our GreenCorps program, if they knew anybody who was a Microsoft Access expert. And I was put in contact with someone named Don Bailey, who works at the RETAP program for the MPCA. And he was very kind to start meeting with me and to discuss project updates and let me just kind of berate him with questions about Access and Visual Basics. And he was a really big help in putting this project together. So for about 4 or 5 months, we met and we would talk about the project and just kind of bounce ideas back and forth. And then I also received a lot of input from Terry and Zach about what additional parameters we could put in, what this might look like in the future, how it might change in the future, what would be good to do for the database to make sure that it's ready for other people to use it, and other inputs such as that. So this is what the database looks like when you initially open the file. We have our site data, our infiltration data right over here, which is that MPD that we talked about. And then this is all site data that we will take on site at each site. So, and then we have these new and delete site buttons, which if you want to create a new site, you press the new button. And then if you want to delete a selected site, press the delete button. And then over here.
That's the form.
Yes, yes. And then I thought it would be pretty overwhelming if we had all of those 67 metrics on just one page at the very beginning. So over here, we have these buttons, which are the other categories of soils data that we collect. And when you click on each of these buttons, these will bring up a pop-up form. And so these pop-up forms kind of break down the data into a bit more sizable chunks, so it doesn't feel like you're so overwhelmed by everything just at the get-go. So these are what the forms look like for our profile, properties, and composition data. And finally, we have a report button in this upper right corner as well. And when you press that button, it will generate a site-specific report, of all of the site data plus the composition, properties, and profile data. So that, which is intended to be used to show, to give to Bar or to show to Terry to talk about the state of a certain site. And then finally, after that, the third project I've been working on has been helping the education and outreach departments. This picture was from a recent visit with Eden Lake doing macroinvertebrate sampling. And then this is a picture from a visit with St. Hubert's where we did an activity rotation. And this specifically was from a birds and bugs activity. So another timeline for you. So a lot of the fall was reaching out to a lot of teachers and kind of discussing options with them. There are a lot of teachers in our district, and a lot of them showed interest. So we were sending a lot of emails back and forth about what topics they were interested in, class and other logistics such as class sizes and dates and what they exactly were looking for, if they were looking for a classroom visit or if they were looking for renting materials or something else. So that was a lot. I spent a lot of the fall doing that, and then also getting familiarizing myself with what the district had, such as the educator grant program, the materials library, and other materials that we had specifically for educators, so that I could present that as an option for teachers. And then from January through May, we continued to plan those classroom visits. And we also started to facilitate about 1 to 3 classroom visits per month, where we would go and do activities with students within the district. And I also helped in creating some lessons plans or adding lesson plans to the materials library that we didn't have beforehand, such as a soil profile lesson. So some of the topics that we have done over the course of my term have been macroinvertebrates activity, which is always classic. This is a profile that one student made from the soil profile lesson. I believe this was from the 4th grade class at Eden Lake. They watched a presentation about the soil profile and then got materials to make their own soil profile craft. And this was a lovely soil profile one student made. And this is a picture from a science night that Eleanor and I tabled at, where we used the Enviroscape to show stormwater runoff and how that affects pollution. And that is my presentation. Thank you so much for listening. Thank you. The next step is to collect more data. We do not have enough data as of right now, and we only really have data from ball fields and residential turf lawns, so we don't really have a comprehensive picture of all of the different ecotypes that are in the district. That's one big reason we really want to do the Arboretum project, is to just get an idea of other ecotypes, because a lot of the soils on residential lawns can be just very mixed up, or just when the property was being made, it just is a bunch of soil that's kind of tossed on there. A lot of the sites at the Minnesota Arboretum are prairies that haven't been disturbed in, let's say, like 40 years or something like that. So the profiles and infiltration and samples are going to look very different. And so we want to kind of get an idea of that as well before we make any really big conclusion. Yeah. Yeah.
So all of this data, or anything that you discovered, or adding data to, kind of, oh, that's surprising. How that wasn't expecting that, or maybe the other side of that is somebody else take that, they figured out anything do unusual.
If anybody has taken the data we've collected so far, and if anything has been. Do you mean just in terms of like other organizations who have done similar studies to this, or?
With any surprises?
Any surprises?
I mean, so you got all this data.
Mm-hmm.
I was wondering back to my something, data pointing somewhere.
Yeah.
The chance to
So it's interesting because some data has kind of been like exactly like we expected. When I first started, we did a project where we sampled a lot of the ball fields from the city center park on Market Boulevard. And I mean, that was kind of, those tests came out exactly like we thought. Like the compaction was awful, infiltration was awful, like it was horrific to do the profiles. It was just so hard to dig down because like obviously people have been running all over it for who knows how many years. So that was kind of exactly as we expected. But as I've been doing the lawns, I've been seeing a lot how soil particle variability will affect infiltration specifically. I was at somebody's property who had a more sandy soil, and I put in the infiltration test, and it was done in like 15 minutes. And like you would think that with a turf lawn, it wouldn't be done that fast. And like my initial reaction is like, oh, that should be a lot slower, but it went a lot quicker. So kind of seeing that variability and that like it's not just completely black and white has been very interesting to see. Cool. Yeah. Yeah.
So
Yes, Zach and I are looking into right now how we can decontaminate our like our hiking boots and the clothes that we wear between sites so that we don't transfer anything between sites. We haven't done that yet, but we are planning to do that. Yeah. Oh.
I thought it was great that you. I didn't know. That's
Yeah. Thank you.
those two. Classes, man.
Thank you.
I mean, I'm sure it's a good investment in everyone, but
hmm
very good and very resourceful. You have that.
Yeah. Thank you. Thanks so much. I appreciate it. Yeah, we're kind of looking to go all over. That's why we're planning to look at there are some prairies in the Arboretum that haven't been touched in like 40 years and have been like under restoration for decades. And there are other parts of the Arboretum that really are not as kind of restored, so to say. When we visited two different forest sites, and one forest site had a lot of duff. It was still very much intact. It looked great. And then we went to another site that was incredibly degraded. It obviously was very affected by earthworm populations and like everything like that. And we plan to sample from both of those to kind of look at the difference and see if that does have an effect on anything. So yes, we do want to get like a whole variety of things. We don't want to look at the things that are just perfect, but it's better to look at everything in between.
Have you done any of the, like use the mustard?
No, we
worms?
haven't.
What worms come up? No.
No, we haven't, unfortunately.
It might be fun, be a great video.
That's true, yeah. I'll tell Zach about that, yeah.
You have, I mean, you guys have.
He hasn't told
Yeah.
me about
I
that.
think so. Yeah.
He's told me not to do it. Yeah, I'll ask him.
And one more question is for anybody to answer. Follow the outcome of all. Where's the best place to look? Where's that all go? Not just the district.
Well, I mean, the district will do a report eventually, but beyond that, I don't know. I'm sure Zach has a much better idea.
Yeah.
About that.
He's told.
Have published a paper.
You said
present.
MCEA?
MCA?
Yeah. Just curious, like, here I want to look up what's come out of this.
I would start initially with, we'll do a soil update report that we do that we have done for soils last year, the year before. And then I would think, I haven't confirmed, but I would think maybe Zach or others or whoever's involved in the project would present at perhaps the powwow, which is the Minnesota Association. Go there now. Minnesota Watersheds.
Minnesota Watersheds.
Or the Water Resources Conference, or who knows? I've been involved in these conversations.
I know there's talk about collaborating with researchers at the University of Minnesota to have them help with our data analysis. There is going to be a lot, and they would be able to help with some of that more intricate data analysis that we might not be able to do on site. I don't know how set in stone that is though, but there is talk of that. As far as I know, there aren't a lot of other organizations that are specifically targeting soil health in suburban soils. But I imagine that once we do get a report up and running, and that if they would want to see our data, that we would probably be more than happy to share that with them. I don't see why not, to be honest. Yeah.
don't know that they've done anything.
Yeah,
yet
just when you
either.
talk about limited sample size, that seems like a way.
Right, yeah.
Nice to come up with that. Management practice. But,
Mhm.
Well, thank you, Rachel.
Thank you, guys.